My name is Andrew Schuerger. I am a faculty member in the Department of Plant Pathology at the University of Florida. I joined the University of Florida in 2003. I had the good fortune when I joined to have a lab available here in this facility that we're talking in called the Space Life Sciences Laboratory. It's a research facility that's adjacent to the Kennedy Space Center. There are four labs here that are operated by professors at the University of Florida. So I joined the University of Florida in 2003, and I came from a company, a very good company called Dynamac. They were a NASA contractor, and they were helping to generate and develop these bioregenerative life support systems. So I had a couple of grants with Dynamac that I was allowed to transfer to the University of Florida. And one of the things I wanted to do by joining the University of Florida is build a Mars simulation chamber. I had experience from 2000 to 2003 working for this contractor. I had the good fortune of being able to work with much larger Mars simulation chambers here at the Kennedy Space Center. I learned how to build them, how to maintain them, tricks of the trade, so to speak, and how you do research inside a low pressure chamber. So when I joined the University of Florida, that was the natural thing I wanted to do. And I started then writing a series of grants related to the survival, growth, and evolution of bacteria under Mars surface conditions. It just was a fascinating area of research. Hit me right at the time, and I got several grants funded right away. And so off I went with my own brand new Mars simulation chamber and three grants that required it as an asset for the research. NASA is very interested in protecting the science integrity of a target location. So if we send a rover or a lander to the Martian surface, we don't want to immediately contaminate that local terrain by bacteria or fungi that might be transported as hitchhikers on those spacecraft. That's called forward contamination. So NASA is interested in protecting a target site from the forward contamination from our spacecraft. There is another process, a reverse process, where NASA would like to protect Earth from any possible microbiota or communities that would be on another target location, because we don't know if there's life on Mars yet. We have to send the right equipment and the right rovers and landers to investigate that. But if there is life on Mars, we don't want it to contaminate Earth in any way. That's called back contamination. So I learned about that just very early on in my NASA research funding. And I decided to spend a major effort on my research looking at the forward contamination issue. It's tough to do back contamination right now because we don't have a bacterium or a fungal species from Mars. But we do know what's on spacecraft, what kind of bacteria and fungi and archaea will end up on the spacecraft, so we can study that forward contamination issue. And so I started doing that. And the main theme at first was, will they survive? Will the bacteria that are on a spacecraft survive under Martian conditions? And the answer was yes, as long as they are protected from the ultraviolet irradiation. There's no ozone, or really just a trace amount of ozone in the Martian atmosphere. So most of the very biocidal UV photons, meaning ultraviolet light in the UVC region and UVB region, all of that radiation, which would normally kill bacteria on Earth if we didn't have ozone, those UV photons will reach the surface and they could sterilize this outside spacecraft surfaces. But how fast does that go is unknown. Is it complete? Is the entire spacecraft can be sterilized by UV radiation in a day, in a year, or in a century? There was just not really good data on that. And so I focused on microbial survival, primarily bacteria, in the Mars chamber. What I find so fascinating about science is it's a process of discovery. So you get to ask questions that maybe nobody has ever asked before in the entire world. Nobody's asked this question. And you get to figure something out and hopefully discover something that humanity as a species has never known about in the past. Discover a new form of life, or discover a new process that's never been done before. Or maybe, as it pertains to Mars, maybe actually discover the presence of an extraterrestrial form of life on the surface of Mars or in the subsurface. I mean, that's just riveting to me to be able to do that.